Good morning everyone, my name is Prakash and I am going to introduce environmental microbiology, a comprehensive overview. So mostly I am going to tell about the role of environmental microbiology, environmental engineering, mostly anaerobic digestion. Okay. So before going that, I will just give a small of my introduction. So I did my undergraduate from Bundelkhand University, India. Then I moved to Pondicherry University for my master's in environmental engineering, and after that I did a research work in SVNIT India as a SRF, followed by I joined as a doctoral program in Inha University, South Korea, and then right now working as a postdoc fellow in the same university and anaerobic digestion. So let's start with what is environmental microbiology. So before going into the detail, first let's know the basics of environmental microbiology. What is environmental microbiology? So environmental microbiology is a composition of physiological or microbiological community which are present everywhere in our environment, from soil to water to air, and also in animal and plants. And you know, the most diverse microbiology we have in our gut system. So our gut system also a kind of a environmental microbiology system. We have to understand this. So these are the three main personalities which have done a lot of work in environmental microbiology. Edward Jenner, who introduced penicillin, Louis Pasteur, which gave the pasteurization method, and then Robert Koch for that typhoid and other disease, how it occurs, what are the microbes involved in that. Okay, so moving on further, these are the events of main environmental microbiology development. So you can see that this is the first time when Prakash suggests that invisible microorganism causes disease. Before that, it was thinking that some kind of godly or heavenly things get us disease or something, but we said no, there is some invisible microorganism or organism which cause us diseases like infection and other things. This was the first concept of microbiological events, followed by Jenner introduces cowpox vaccine in 1798, and cut to the first 1881. This is the first major event when we cultured bacteria into our lab. Before that, we have never cultured bacteria or other things in the lab, and then we cultured. This is the first bacteria which we have been cultured, and genome. Like we also know the gene analysis of this. So this is the first bacteria. In right now, currently, we also say that E. coli is the model bacteria in environmental microbiology. Whatever you have to do the test, you first try on E. coli. That's why we call it as model bacteria in microbiology field. And then this is the E. coli is the only bacteria in 2000, first bacteria which we have genome sequence. Like we know all the gene sequences of this E. coli. After that, we did a lot for other microbiologists like bacteria and fungi, but this was the first one. So these are the major events. Cut to the next. Okay. So this is before going into that, let's introduce the type of microorganism. They are categorized into two types. One is prokaryotic cell. So prokaryotic cells, you can see, is a have a lot of other characteristics and body parts. We can say organelles. So here these are the prokaryotic cell which consists bacteria and archaea. Okay. So there, if you see, they don't have nucleus. They have roughly a small nuclei here. And then the plasmids, the cytoplasm. I will tell in the next slide what is the function of all these small things. So bacteria and archaea, you have to remember, these are prokaryotes. Human body cells are not prokaryotes; those are eukaryotes. So we can say that our cells are eukaryotic cells. Okay. In the next, so what is the function of that? Then each organism have their own function. Like plasma membrane, you can see in the previous one. This one, the green color. It's act as a barrier, like we have a skin. In bacteria, they have a plasma membrane which act as a barrier from the outer environment. Can you use the touch to slide? Okay. Sorry for the disturbance. So the plasma membrane, then we have a gas vacuole. If you see, the gas vacuoles are small things. These are the gas vacuole. Gas vacuole, the purpose of gas vacuole is that they give the buoyancy to the microbes. if they live in the water, because they definitely live in the water surface, so in water they give their buoyancy, like they can float on the water, otherwise they will drown into the water. So this gas vacuole give the buoyancy because it's full of air, there is nothing inside. Then we have ribosomes, these are the protein synthesis. Then we have nucleoid, like we have nucleus in our cell, so they don't have nucleus properly, so nucleoid. And the nucleoid is the thing where all the genetic material for the bacteria stays. Okay. Then we have a cell wall. After plasma membrane, then we have a cell wall. And then slime layer. The purpose of slime layer to attach any bacteria on the surface of like solid or soil. So it helps to adhere with some kind of epoxy. We can say EPS. And then we have a fimbriae and pili. These are the bacteria which they use for the mating and attachment to the surface. Finally, flagella, which use the movement of the bacteria from one place to another. And then endospore, survival. Endospore is very important in bacteria because when under harsh condition, they make a coating of above their surface so that it will not be affected. Like it's a kind of egg cell that is very harsh and rough. So endospore, the function of endospore is that one. And here it comes to the eukaryotic. So all the plants, animals and fungi is so they have eukaryotic cells. They have a proper nucleus. You can see here, in which all the DNA and RNA we have here, and then we have a cell surface. We have a flagellum. We have a plasma membrane. So almost 80% of eukaryotic and prokaryotic cells are similar, but some are different. Like they don't have mitochondria. We have mitochondria here. Mitochondria we consider. Sorry, here mitochondria we consider as a powerhouse, which gives us the power to move, to cell to function. So these are the mitochondria. They have their own nucleus and genetic material. So this is the small introduction about the eukaryotic cells. Again. But for the bacteria, it is around 1 micrometer, and for the virus, it is in nanometer. Okay, so these are the size comparison of each organism in the cells, like flu virus, protein, atom having 0.1 nanometer, and compared to the frog egg, here you can see it is a 1 millimeter. So we survive in this range, and bacteria having this range. So it's better to remember these values and size. So it will, in the future, you will have a help of this. Next, come to the typical composition of bacterial cell. So in bacteria, mostly if you consider what are the major things, we have protein 55%, polysaccharide 5%, lipid 9.1%, DNA 3, and other things are the rest of things. In elemental composition, we have carbon 50%, oxygen 22%, nitrogen 12%, and then rest of other are in the 1 or 2%. So here we are considering the constituents of cell, and here the major elemental composition of cell. Okay. Now fundamentals of microbiology. For the cells to function, they have one thing, energy, another thing is carbon, and then inorganic material like some kind of nutrients. So energy is coming from different sources. Different bacteria use different sources for energy, and for carbon also different bacteria use different sources of energy. I will come to that in the next slide. So this is the organic, what is the synthesis reaction for the microbes? These are the equation. Organic material plus oxygen plus ammonia plus phosphate utilized by microbes gives new cells. So new bacteria will develop on biomass, we can call it, and then release CO2 and hydrogen, water. Okay, and then in some microbes they also utilize this nitrate and nitrite and release nitrogen gases. Okay. Here are the two common sources of carbon. So now we are going to classify bacteria in the terms of their utilization of carbon energy sources. So the two common sources of carbon are carbon dioxide. So those bacteria who use carbon dioxide as their carbon source called autotrophs. Those bacteria who use organic matter used by heterotrophs as a carbon source are called as heterotrophs. So I will tell you all those photosynthetic bacteria which uses carbon dioxide, like plants and other photosynthetic bacteria, they are autotrophs because they use carbon dioxide as a carbon source. But heterotrophs, all those pathogens which are disease causing bacteria, they use organic matter as a carbon source. So we are also an organic matter. So for us they are heterotrophs. So we have divided in terms of carbon. Now comes to the energy. Those bacteria who use light as an energy are called phototrophs. Okay, and those bacteria who oxidize organic and inorganic compounds are called chemotrophs. Okay, so all bacteria which uses light, like algae, those are phototrophs, and other bacteria like pathogens, they use oxidation of organic and inorganic compounds are called as chemotrophs. Now the third one, the most common source of electron. So all those bacteria who reduce inorganic molecules, not organic, inorganic molecules like metals or some kind of other inorganic molecules, they reduce and get the electron. Those are called lithotrophs, because lith means lith means actually I think Latin or German term, which means stone. Okay, so it's inorganic, so used by scholars lithotrophs. And those bacteria who use electrons from organic molecules are called as organotrophs. So these are the classification we have done in microbiology based on their uses. What are the source of electron, what are the source of energy, and what are the source of carbon. Okay, so here I have made a table to show what are the sources and what kind of bacteria fall into this. So nutritional type, photo means light, litho means inorganic, auto means carbon dioxide, not organic. Sources of energy, light, inorganic, and CO2. So what are the bacteria? Algae, purple and green sulfur bacteria, and cyanobacteria. So these will be fall into this category, photo, litho, autotrophs. Okay. Now photo, organo, autotrophs. So there are a lot of bacteria who use energy from light, but they need carbon from organic sources. So they will be called heterotrophs. So photo, organo, heterotrophs, light, organic and organic carbon sources. Which bacteria? Purple and green non-sulfur bacteria. Okay. So these are non-sulfur bacteria, so they use organic carbon for their electron. Then chemo, litho, autotroph. So chemo, chemical energy, litho, inorganic, and autotroph, light energy. Okay. So autotroph, CO2, carbon dioxide. So here sulfur dioxidizing bacteria, hydrogen producing bacteria, nitrifying bacteria, and iron oxidizing bacteria are fall into this chemolithotrophic. Then chemo, organo, heterotrophic. The same way, chemo, chemical energy, organo, organic, and hetero means organic carbon source. So non-photosynthetic bacteria and protozoa fungi are chemo, organo, and heterotrophic. Here pathogens will also fall in this category, all the pathogens. Okay, because they are chemo, organo, heterotrophs, and some will fall into photo, organo, heterotrophs. So coming to the next, what are the energy sources they have? Aerobic heterotrophic nutrients, organic compound, bacteria energy synthesize, they require oxygen and nutrients from other sources, and then new cell will form, CO2 and water. Aerobic autotrophic, organic compound, oxygen, and then they will produce new cell and CO2 plus H2. So these two are almost same. Okay, but in the second one, anaerobic condition, heterotrophic, organic compound, oxygen, nutrients, new cell, and they will produce CH4 and CO2. So these are the microbial energy sources. Heterotrophic bacteria metabolize biological oxygen demand and try to go in the biological process. So this is oxygen, we are not saying that they are consuming because they are anaerobic, so they are not taking oxygen. But why we gave oxygen here? That how much oxygen is required to oxidize this organic compound, but here we convert into CH4. So to measure the COD and BOD, which I will tell you later, we calculate how much oxygen is required. Okay. So here don't confuse with that anaerobic bacteria utilizing oxygen. I just put to calculate the BOD. Okay. Now electron requirements. So there are two kinds of bacteria which survive into two kinds of atmosphere. One is anoxic atmosphere and one is anaerobic atmosphere. Anoxic atmosphere is what are those? No free oxygen but oxidized compounds like nitrate. So in anaerobic condition, if you see here, oxygen is the electron acceptor. Okay, but if the oxygen is not there, then who will be the other electron acceptor? Nitrate, sulfate, and other things. Okay, so that atmosphere will be called as anoxic, and then in anaerobic, no free oxygen and no oxidized compounds. are present. So in this, no oxidized compounds will be present also. So no nitrate here and no oxygen. So that atmosphere will be called anaerobic atmosphere. Okay. Here you can say aerobic organic compound oxygen synthesis 0.45. This is the highest one. That's why you have seen in the wastewater, whenever there is aerobic treatment, they have a lot of sludge. Those sludge are representative of the yield, like biomass. But if you see in case of anaerobic, it's minimal. Okay. So that's why in wastewater we say that in anaerobic condition we have a lot of sludge production, but not in anaerobic condition because of this thing. Next, if you go, then microbial classification depending on the carbon source and energy uptake mechanism. So in autotroph, an organism that produces complex organic compounds such as chemoautotroph and photoautotrophs. Actually here it is distorted, but it is the same. This you can see this algae, CO2 plus sunlight plus nutrient gives cells like algae cells that they form. Okay. In the next, I will show you again the microbial classification depending on the carbon source. So here the carbon source is suppose acetate or glucose. In aerobic condition they produce hydrogen, CO2 and cell. This is aerobic condition, but in anaerobic condition they produce CH4 and CO2. So glucose to acetate and acetate to CH4 and CO2. So this is in terms of anaerobic condition. Okay. Next I will show energy balance. So in terms of wastewater treatment, either aerobic or anaerobic, it is very much necessary to have energy balance. Like which kind of energy? Because we know that energy can never be created nor be destroyed. So here organic energy which is converted into either biomass or methane. So we have to calculate how much energy is going each way. So if we have a COD, some organic content, 50% is going into CO2 plus H2O in under aerobic condition when oxygen is present, and 50% is going into sludge, a huge part of sludge. But in case of anaerobic condition, if you have some organic matter, 90% is going into biogas and 10% going into biomass. So you can see the difference, 40% difference, and that is the benefit of anaerobic digestion that I will talk later. Here, energy generation aerobic versus anaerobic. So from energy perspective, let's see which one is better. Glucose, aerobic condition, CO2 and H2O, 2870 kilojoule per mole, and that is negative. So this releases a lot of energy. It means this reaction is very much spontaneous. But in case of anaerobic, you can see the value is minus 390. So here energy is not that much produced, but here we need this one. So this is as a part of energy we can consider. Okay. And then in terms of ATP, in aerobic we have a huge ATP generation 34 to 36, but we cannot utilize this because it goes into the biomass formation like sludge. But in case of this, we can utilize it in form of CH4 and low biomass formation. So here large cell production 34 to 36 ATP and low substrate, but here high substrate and 2 to 4 ATP in case of anaerobic condition. Residual energy gaseous, like in the anaerobic condition we have methane, hydrogen sulfide. Another acid we have short chain fatty acid, medium chain fatty acid and alcohol, and electricity and lipids. So these are the benefits we can get from the anaerobic condition. Next, what is ATP? ATP is we can say is the energy currency of human body. Like if we have to go somewhere, if you have to eat something, we need money. Okay. The same way, if our body has to work somewhere, like if I'm talking like this, it's consuming ATP. So all the energy in our body, from thinking to walking, everything comes from the ATP. What is ATP? This is the chemical formula of ATP. You have seen that 3-phosphate ion has been arranged here, and this is the carbohydrate, and this is the adenosine thing. So the full form is adenosine triphosphate for ATP. So this is the currency, and when we have to utilize something, every time one phosphate will release. So suppose if you are going to consume ATP, so one phosphate will be gone from here, and then it will be diphosphate and some form of energy will release. Again we need more energy, then one more phosphate will go, and then it will be adenosine monophosphate (AMP), and this will, which every release of phosphate we will receive some kind of energy. Okay. So here I will show you in aerobic respiration and anaerobic respiration, ATP is forming to ATP, but because we need to gain also, that's why we eat so that ATP is formed. So in terms of fermentation, photosynthesis and respiration, ADP is coming to ATP. Again we are getting the balance, and then we are doing some work like walking, speaking, giving lecture and swimming and all the mechanical work. ATP is breaking down into ADP. And how much energy we release? So with every phosphate ion release from this, like with one phosphate ion detached, it is minus 30.5 kilojoule per mole energy is released. Okay, so we gain this much energy. Other things are also there for this NADH. Those are other secondary energy currency, we can say that. NADH and FADH, those are also formed, but the main is ATP. So 32 kilojoule requires to make 1 mole of ATP. So if we have to synthesize ATP in our body, we need 32 kilojoule of energy. Okay. Now come to the ATP synthesis from glucose. So this is the pathway how the ATP is synthesized. So first, when we take some organic content like glucose, so glucose goes under glycolysis, okay, and then it forms 2 pyruvate, 2 ATP and 2 NADH. So here we got 2 ATP. Then it comes to the next Krebs cycle, we also call it TCA cycle, occurs in cytosol in our body, in our cell actually. No oxygen required, this is no oxygen function process. In this 3 NADH, 1 FADH2 and 1 GTP is formed. If you need what is FADH, it's a flavin adenosine dinucleotide. It's also kind of a small energy will be produced if from the breaking down of FADH2 to FAD. And then GTP is also a small currency for the energy. It's called guanosine triphosphate. So there are a lot of other things we have in our body to generate energy and then consume again. Occurs in plasma membrane. Then we have final one, electron transport chain. That occurs in plasma membrane in prokaryotes. Oxygen is required, aerobic respiration, and in case of anaerobic respiration, other inorganic ions are required if oxygen is not present. Now let's calculate that how much in each pathway have. In glycolytic pathway, as I mentioned here, glycolysis. Here in glycolytic pathway we generate 2 ATP, okay. In oxidative phosphorylation 6 ATP. Then in the pyruvate, here the pyruvate, here we generate 6 ATP. Then Krebs cycle 2, 18, 4. So in total in aerobic condition 38 ATP has been generated, okay. However, there are some problem because this process is not 100% efficient. So we get only 2.5 and 1.5 from NADH, and if we get the ratio, the total ATP yield is 30. So from 38 to 30 reduced. Now go to the anaerobic condition. So in anaerobic condition, how this reaction happens and what are the conditions, because in anaerobic condition there is no oxygen, so the electron acceptor is sulfate, nitrate, Fe3 plus, and this is I just made to show you the in case of oxygen. So these are anaerobic and this is aerobic condition. So you see how much electron volt has been generated. This is negative 0.42, but here is plus 0.82. So this is more spontaneous compared to each other electron acceptors. Okay. So this is the difference between, you can say that which one is having higher affinity towards electron. So the more positive, the more higher affinity, the less positive, the less higher affinity. So since oxygen has a higher affinity, then it has a 0.82, and NADH and hydrogen has a less affinity, then it's minus 0.42. And again, ATP generation in aerobic condition will depend on the source of electron acceptor. So also, if oxygen is we are using as electron acceptor, then the ATP generation will be different. But in case of these other electron acceptors acceptance, the ATP generation will be different. So which I showed here, 38 or 30 is based on the oxygen or aerobic respiration. Now come to the growth of microorganism. So these are the basic which I have explained. Now these are the growth factors which affect the microbial community, mostly bacteria, because we are going to focus on bacteria from now on. So the first is pH. So those bacteria we have categorized which one is which bacteria like which kind of atmosphere. So if a bacteria can grow in a pH of less than 5.5, 0 to 5.5, we call it acidophile. Phile means loving. So those bacteria who can grow below 5.5 are called acid lovers. So, and those 5.5 to 8, those are called neutrophiles. Those are like neither more acidic nor more basic. Okay, and then those bacteria who can grow from 8.5 to 11.5 are alkalophiles. So in anaerobic digestion, when fermentation happens, mostly are acidophile and neutrophiles. But for methane production, mostly are above like 8. They can grow easily in alkalophiles and in this range 7.5 to 8. Okay. Now come to the temperature. Again we have categorized bacterial atmospheric condition into 3 categories. So those bacteria who can grow easily well below 15 degrees Celsius, 0 to 15 degrees Celsius are called cyclophile. Cyclo means very low temperature and phile means low temperature loving. Mesophile 20 to 45, and then here thermophile above 55 degrees Celsius. So these are the categorization of bacteria based on their growth. Now come to the oxygen. Here you can see there are 5 categories, 1, 2, 3, 4, 5, and it's based on the how much oxygen they can tolerate. Okay, so this is obligate aerobe. So obligate aerobes are those aerobes, they grow only when oxygen is present. If oxygen is not there, these bacteria, these microorganisms will not grow. These are called obligate. Obligate means obsolete. Okay, absolute aerobe. Now there are facultative aerobes. Facultative aerobes are those bacteria, they love oxygen to grow, but if oxygen is not available, then they can also grow. But if oxygen is available, they will prefer oxygen as an electron acceptor. So you can see here in obligate aerobe, this is a test tube, okay, filled with their medium, and this is the aerobic part. So all these bacteria are very much dense here because it's contact with air. Nothing is here, right? In facultative, you can see the bacteria are here but not very much concentrated. So near oxygen they are very much dense, but if oxygen is not present, they are growing but not very much good condition. Now aerotolerant aerobe, or those bacteria who can tolerate air, little bit of air, but they don't want much air. So you can see that they are grown evenly in the test tube, but they are not growing, they are not dense here, they are not dense here. Okay, so they don't like much more oxygen, they don't like less oxygen, they are in the middle of that. Okay, little bit of oxygen is okay, but more oxygen they will die. Now strict anaerobe. Strict anaerobe are those microorganisms, those who can grow only when oxygen is not present. You can see here oxygen is available on the surface because it's contact with air, so no bacteria growth. Here all the bacteria are growing, right? And then another one is microaerophile. Microaerophile, they grow in anaerobic condition, but they need little bit of oxygen. Okay, just little amount of oxygen to grow better. So you will see that they are they don't grow here, they don't grow on the top, they are in the middle of that. Here because as you go down, oxygen concentration is lowering down. So they said we don't need we cannot grow here, so in the middle they have more dense growth. So there are 5 categories of the bacteria: obligate, facultative, aerotolerant, obligate, anaerobe, and then microaerophiles. Okay, now I will tell you microaerophiles can tolerate 2 to 10% of oxygen for their growth, but if you provide 20% of oxygen to them, they will die. Okay, so most of the anaerobic digestion process, the fermentation bacteria mostly are facultative anaerobe, but methanogens are strict anaerobe. Okay, so in an anaerobic condition we will not have this, we will have this, and we will have this one. Okay, these are bacteria who do the fermentation, these are archaea who do the methanogenic process. Now come to the classification based on their structure. So you can see the root of the tree for bacteria, archaea and euarchaea. So methanogens, those methane producing bacteria, are not real bacteria. So we have made another branch for them. Those are called archaea. There is a slight difference between a bacteria and archaea, I will tell you later. So bacteria, one branch, all those bacteria can grow. Here archaea, one branch, animal, fungi, plants, and then one branch which is more leaning towards a euarchaea, but they are little bit tilting towards bacteria. So those are methanogens, you can see. Methanogens are archaea, not bacteria. So there is a slight difference in their structure. Now I am going to show the bacteria and their shapes. So what are the shapes of the bacteria? So bacteria are single cell, utilize soluble food efficiently. Okay, consider primary feeder in a wastewater treatment plant. So these are their shape: spherical shape or cocci shape, round like a sphere. Okay, so if any bacteria whose name has ended with cocci, you can say that this bacteria structure will be spherical. Okay, if any bacteria whose name ends with bacilli or start with bacilli, then their shape will be like a very long shape, cylindrical shape, you can say. And those bacteria whose name end with helical or spirally, they are mostly like hair strands. So these are the majors. There are more too, but those are not very much discussed and not very much dominant. So these three shapes of bacteria are very much dominant. Okay. So all the bacteria who use in the fermentation, either they are spherical or cocci. And in methanogens also mostly they are cocci. I will show you the name. See here in archaea, Methanococcus, their shape is round, but Methanobacterium, their shape is spiral form. Okay. So here I will tell you some names. If you had heard about bacilli, some bacteria end with bacilli. So it means their shape is this one. Have you heard about this pathogenic bacteria Streptococcus? Their shape is round because their name is stand with coccus. Okay. So with the name also you can tell that bacteria has either this shape or this shape. Okay, here how about archaea? Okay, I will tell you the difference in bacteria. Bacteria are single cell, utilize soluble food is efficiently, and they have these shapes. In archaea, single shape, but in their cell wall they have peptidoglycan. This you will not find in bacterial cell wall. So in archaea they have pseudopeptidoglycan. In bacteria we have peptidoglycan. Okay, there will be a little bit of chain difference. Then in case of archaea we have ether linked in cell membrane, but in bacteria we have ester linked. Replication origin is multiple, but in bacteria it's single. Alanine polymerase multiple, modified TCA, Krebs cycle. So here which I showed you Krebs cycle. Here which I showed you Krebs cycle. This is strictly followed in bacteria, but in archaea it is not completely followed. All the steps. They cut the steps and they form their own processes for the ATP generation. So they have a difference. These are the differences between archaea and bacteria. Okay, so I will tell you one thing you have to remember. All the methane producing bacteria are not bacteria, they are archaea. A bacteria never produces methane. They are always produced by archaea. Okay, so if somebody will tell you that bacteria produces methane, completely wrong. It's archaea produces the methane. Bacteria never produces the methane. Okay. Now come to the growth pattern. So this is the general growth pattern for the any bacteria or archaea. This is the lag phase, then log phase, then stationary, and death phase. I will explain what are these things. So lag phase, when you culture the bacteria and put some kind of organic matter. So in lag phase, what they do, they recognize the organic content, what are those things, and then according to that they synthesize some chemicals in their body to consume it. So by that time you will not see any bacterial growth. So they are understanding what they have given to feed. So lag phase. Okay, if you read here, lag phase are adaptation of the cells to the new environment or new organic content, and after that they will start to the log phase or exponential phase. So in this phase, once they recognize, okay, these are the things we have to adjust it, they adjust it, and then they start growth. They start consuming, replicating their population and all that. So it's called exponential phase. So here the bacterial growth happen in a like 2, 4, 8, 16, 32, like in that way, because they always grow into 2 to the power n. Okay, and then the stationary phase. In stationary phase, what happens? The resources are limited, so the bacteria growth is equal to bacteria death. Like, because we don't have much organic matter or food left for them to eat, so whatever they are eating and growing, because some bacteria also they produce some kind of metabolites to release toxins, because there is not much food. So what they do, to get more food for themselves, they release the toxins, so the other bacteria dies, like kind of a cannibalism. Okay? So this is stationary phase. So death and birth will be equal. Okay, and now in death phase, when nothing is left, all the toxins are there, so no bacteria can survive. So now it's a death phase. Okay? So this is how happens for the bacteria.